we've got an installation which is being built by an organisation called Makerversity, which is based in Somerset House, and we're working with them and the Cultural Institute at King's, constructing an object or an installation that will actually produce sort of 3D printed bits of DNA that will sort of accumulate during the festival and make something much larger, and that will involve audiences interacting with the installation and telling it what sort of DNA to produce.
So the DNAted project aims to create an urban sculpture through which people will be experiencing a bottom-up approach towards making artifacts using the DNA strands. Every person who comes and enters into this interactive installation, in a way, would come and get educated about fundamental principles as to how the artifacts or natural organisms, in fact, are created from DNA, and then we draw parallels as to how we are translating this information into an art project and creating this sculpture with contribution of more than 200 people. Throughout the process, Arts and Humanities, as well as the Cultural Institute, put me in touch with scientists who were keen to collaborate on an art project, and I met Professor Brian Sutton, and we formed this amazing collaboration as to how we are able to take meaningful concepts from biological organisms and apply them in design.
Well, DNA stands for deoxyribonucleic acid, which is best thought of as a very long molecule in the nuclei of every cell in our body, and indeed the nucleus of every cell of every organism. It's segregated into chromosomes, 46 in the case of humans, but best thought of really as one long molecule. And we have a model of that here. It's a long chain polymeric molecule consisting, in fact, of two chains wound around each other in a famous double helical structure. But the important aspect of the molecule is that it has a base, a chemical entity, of which there are 4 types in DNA, in all DNA in every organism on the planet. Either adenine or guanine, cytosine or thymine, A, T, C, and G. And the remarkable aspect of the molecule is that it contains a code consisting of these letters. So we have this direct relationship between 3 bases and 1 amino acid. So in this project, you will have the opportunity to just invent a base sequence, and we'll let you think of 9 characters. So C, G, T, A, until there are 9 characters. Now you now know that those will be read 3 at a time. So those 3 triplets will correspond to 3 amino acids, if you like, but in our case, these are 3 building blocks of a sculpture. So just as proteins, linear strings of amino acids, once they have been synthesized in the body, fold up to generate the enzymes or the antibodies or the structural proteins, whatever it happens to be, in skin and bone, these 9 bases will translate into 3 amino acid building blocks of the sculpture, and they will fold in a particular way, and we've ensured that the way they fold, the way they pack against each other, is determined not only by the nature of the 3 amino acids and therefore the particular code that you wrote in the first place, but also the relationship 1 amino acid to the next, and that will determine how the next one folds. So there is a direct analogy between DNA coding for the construction, if you like, a blueprint for constructing an organism or a human body, and the sequences of bases that you've been typing in, and to generate unique structures that lead to the formation of these unique elements within the sculpture.
So the theme, fabrication, feels like it's made for us because we are a makerspace, we're a place where people are based to make things. It's what we get excited about. We get excited about it in a very particular detailed way. We get excited about process, we get excited about tools, we get excited about machinery and materials and playing with all of those things. What's really exciting about it for me is bringing together a kind of world of ideas and critical thought together, I guess, with a world of technology and production, and actually showing how those 2 things together create something that is much more interesting. So a lot of the people who work here, for instance, when you say to them, why do you make? They'll say, because it helps me realize an idea. And I think in the arts and humanities world, thinking about ideas and realising those takes a very different form. So I think it's really exciting to do something where it takes a physical form, and to do that using, this new mythical technology of 3D printing that people talk about the whole time, to create something, really amazing with real depth, with real kind of insight behind it.
So DNAted is a site-specific participatory installation based here on the Quad King's College campus. We invite you, the public, to come along and to be part of the DNA process, and to talk to the team at Studio Integrate, who we've commissioned this year for the commission as part of the Arts Humanities Festival of Fabrication. So we put an open call out and invited applications from designers and makers based here, and people responded particularly to DNA, partly because the site of King's College and the site underneath the quad where the installation will be sited is very important in history because of the discovery of DNA that led to DNA from Maurice Wilkins and Rosalind over 60 years ago.